Everyone, I think we're going to go ahead and get started. My name is Christy Sullivan. I'm the Secretary for the American Society for Cellular and Computational Toxicology. Thank you for joining our webinar today. Just briefly to tell you a little bit about the ASCCT, we are a scientific society that aims to provide a platform for learning and discussion and collaboration about in vitro and in silico methods for toxicological testing. We have a yearly meeting, and that meeting is in the fall. And I'm going to go ahead and tell you that our next meeting has been scheduled. It is September 29th and 30th, 2016 in Research Triangle Park in North Carolina. And we are happy to have confirmed our first plenary speaker. It's going to be Dr. Thomas Hartung from Johns Hopkins University CAT, and he'll be talking about read across. We don't have a title yet, but then what we're going to have is a first day of sessions on a read across kicked off by that plenary, and also other general talks from submitted abstracts. We always have a very nice poster session and our business meeting, of course, and a reception. And then we'll be having a second day, about half to three quarters of a day, with a theme of pluripotent stem cells and predictive toxicology, with a panel organized to discuss that topic. So we're really excited to start planning this meeting and hope you will consider heading over to our website in a few weeks' time to learn more about the meeting and submit your abstracts as well. So let me go ahead and get started with the program for today. First of all, I want to let you know that this webinar is recorded. ASCCT members will have access to the recording after the event. I've put in as a handout for this webinar the PDF of Dr. Lawless' slides, so you can download that if you would like to. After the presentation, there's an opportunity to ask questions, and so please put your question in the question box down there, and we'll get to them after the program. And you can also reach me if you're having any trouble via direct message in the chat in the lower right-hand corner as well. So without further ado, I'd like to welcome both Dr. Michael Lawless and Dr. John DiBella. Dr. Lawless is a business development manager for Simulations Plus for Cheminformatics, and John DiBella is VP for Sales and Marketing. They are co-authors on the presentation, and will be Dr. Lawless will be giving the main presentation, and John will be talking more about during the question and answer, likely. So let me go ahead and get started. And again, the title for the talk is Integrating in Silico Predictions with PBPK Modeling.
Okay. Thank you very much, Christy. Before we get started, I want to thank Christy Sullivan for organizing the webinar and giving me the opportunity to present some of our research. I also want to acknowledge and thank my co-authors, John DiBella and Mike Bolger. And of course, I want to thank everyone for attending this webinar. There are two sections of the presentation: predicting oral bioavailability from 2D structure and using in silico modeling to predict the renal failure caused by SGX 523. So we recently, in the past few years, have developed models to predict cytochrome P450 kinetic parameters, notably KM and Vmax. And these models can be incorporated into PBPK simulations. And so I was interested to see how well we could predict oral bioavailability. Thus, I looked for compounds with experimental oral bioavailability and at a given dose, and whose major clearance pathway is CYP-mediated. So that's kind of the overall subject of the talk. Now let's look at what's happening in vivo. So if you take a pill, a drug, it will enter your stomach and begin to dissolve. So that's the D, the dissolution. So some of the models you would use to simulate the dissolution would be solubility, both aqueous or water solubility, and biorelevant solubility. Biorelevant solubility would be the solubility in simulated gastrointestinal solutions. And then also the pKa of the compound. In the stomach, the pH is around between 2 and 4, let's say, and so if the compound is ionized, that's going to affect its solubility and its dissolution. Also, the diffusion coefficient is a parameter we would predict. Once the compound is dissolved, it can start being absorbed into the gut wall. So the next step would be absorption. And right as the compound enters the gut wall, this is termed the fraction absorbed, or FA%. Things that might influence the FA% are the log D of the compound and the permeability. With the PBPK simulations, we're able to model both transcellular permeability, which would be the compound going in one side of the cell and then coming out the other, and paracellular permeability. So very small molecules can actually fit between the cells and go into the gut wall, and diffusion would be another parameter. Now, there are enzymes in the gut wall that can metabolize the compound. Here I've just listed CYP kinetics, but also there are UGTs in the gut wall. Now, I'm not emphasizing that in the talk, so I just listed the CYP kinetics here. Next, the compound will start traversing into the portal vein. And this is termed FDP. Now, if FDP is lower than FA, that indicates that some metabolism occurred in the gut wall. So now the compound's being carried or transported via the blood, so you need to pay attention to the fraction unbound to plasma proteins, and then the blood to plasma concentration ratio. The compound will then enter the liver through the portal vein. And again, this is where other CYPs can metabolize the compound, so we can account for those. Then the compound would go into systemic circulation. This is termed the oral bioavailability. So when we compute this, we're looking at a mass balance equation. So what was the amount delivered? What's the fraction that comes out on the other side of the liver? Experimentally, you would measure this by taking the ratio of the area under the curve from an oral dose to the area under the curve of an IV dose. Not all compounds have a reported oral bioavailability, and in some cases it's because the compound has too low a solubility to deliver it intravenous, by an intravenous route. And then there's a lot of parameters, obviously, we get out of the PBPK modeling. One is the plasma concentration versus time. We'll talk a little bit more about that in the second section. So this is just an overall scheme of the modeling and the presentation. So first we created a database of compounds with a fixed dose or a known dose and their oral bioavailability at that dose. It's important to include the dose because the oral bioavailability is dependent upon the dose in some cases. If it's a very low soluble compound, it might be the oral bioavailability might be dose limited, and so as you increase the dose, you can possibly decrease the oral bioavailability. Most of the compounds, the oral bioavailability was obtained from Goodman and Gilman's, the pharmacological basis of therapeutics. Others I got directly from the drug data sheet along with the dose, and then some doses I just got out of websites like drug.com. The next thing we needed was the major clearance pathways. I mentioned we wanted CYP clearances. So there was a paper published in 2014 in Drug Metabolism and Disposition by Tashimoto, where they were trying to predict the major clearance pathway of compounds that were cleared renally via some CYPs and via transporters. And so they had a nice table in there with major clearance pathways, and I tried to extract ones where that was CYP mediated. The way they define the major clearance pathway is that it is considered to contribute greater than 50% of the total body clearance. So it's not 100% that it's cleared through the CYP, but more than 50%. So I found 62 drugs. I certainly think this can be expanded, but I was kind of doing this one at a time, kind of checking all the input parameters. Next, we'll compute various properties such as solubility, permeability, the CYP kinetics, and here we used ADMET Predictor 7.2 to predict those. Then we use that as input into PBPK simulations. Here we use GastroPlus to perform the simulations, and our physiology was for a 35-year-old American male. A number of outputs are created from the program. Excuse me, one is the, and we basically looked at FA, percent, FDP, and oral bioavailability, and then compared the oral bioavailabilities back to the reported values. This slide just has some examples out of the data set. What I was trying to do was look at kind of the range of oral bioavailability. So we started low of 3.1 from flufenazine and go up to 85% just in these examples. For ibuprofen, also included a dose here. Ibuprofen is the largest dose at 400 milligrams. And then the major clearance pathway is also reported. This is just kind of a broad look at the data set. The oral bioavailability values range from 3 to 99% with an average of 60%. There are some compounds where either you had a standard deviation associated with the number or you had a range, say 10 to 25. And for about 43 of those drugs, there was more than one reported value. And the CV percent, which is the standard deviation over the mean and shows the extent of variability relative to the mean, was 29%. So, even experimentally, it's hard to get an exact number for the oral bioavailability. The molecular weights range from 206 to 914. This is a pretty large compound for a drug. Typically, drugs would be under molecular weight under 500. The highest log P was 7.5. The lowest was about 1. Again, and this is a predicted value. On this line, I showed the log D at pH 7.4. It ranges from about 0 all the way up to 5.4. So this compound here obviously has some ionizable groups, which reduce its log D at 7.4. For the solubilities, they range from a low of 2 micrograms per milliliter all the way up to 9 milligrams per milliliter with an average of 0.56. There were 10 acids, 28 bases, and 24 neutral compounds in the data set. 10 of them had a major clearance pathway at 2C9, 15 were 2D6, and 37 of the molecules were 3A4. And then down here I just have a distribution of the reported oral bioavailability. Typically, you'd want more than 50% oral bioavailability when you're designing the drug, but there are some down below 20%. So we're going to create models and then use those models in PBPK simulations. So I just want to point out this fact that, or this quote from George Box in 1979. All models are wrong, some models are useful. So we can't represent nature perfectly with equations. So we make assumptions and approximations, and our goal is to have reasonable, not perfect models. But reasonable does not mean poor. And we need to have the same expectations for simulation and modeling and put those in the same light as wet lab experiments. Okay. So you got to keep that at the back of your mind. And this is just some of the models we used in the PBPK modeling. So the top 3 or top 4 are related to solubility, log D, pKa. We have a very large pKa data set, permeability, fraction unbound, as a percent here, and then these SIP kinetic parameters, which I'll talk a little bit more extensively about. So this just lists 3 keys to creating good QSAR models. You need good, clean, and consistent data. You've heard the adage, garbage in, garbage out. You also need good, appropriate, and accurate descriptors, and I'll address that on the next slide. And then finally, you need good training algorithms. And the training algorithms are going to relate the biological activity to the predicted values. So if we're looking at a bulk property such as a log P model, the molecule goes from the water to the N-octanol, and it's the ratio of those 2 concentrations. So bulk properties such as this depend upon the whole molecule. The molecule is not undergoing a significant change, and the interactions are non-specific. So in this case, whole molecule descriptors are okay. So a whole molecule descriptor might be like the molecular weight of the molecule, or the volume of the molecule, etc. When you're looking at an enzymatic reaction, for this example, we're looking at a SIP. So an oxygen atom is inserted between the CH bond of this phenyl ring. So the interactions are specific. There are certain atoms that are more important than others. Obviously, this carbon atom that's being hydroxylated is more important for this particular reaction than this alkyl carbon in the ring. The molecule does change most of the time. So in order to model these phenomena, we need descriptors of reactivity. So we created an EEM Huckel charge model. EEM stands for electronegativity equalization method, and we parameterized these methods to reproduce ab initio charges. So we took about 2,000 to 3,000 molecules, performed high-level ab initio calculations on them, and then extracted the atomic charges for each one of those, and then we parameterized the EEM Huckel combination method to reproduce those atomic charges. And so I realize this graph's a little small, but our correlation coefficient was 0.9858. So we're accurately reproducing those, and we can very quickly calculate these values. Since we're doing molecular orbital calculations, we can get reactivities out of these, such as Fukui indices and frontier orbital atomic densities, and that relates the reactivity of that particular atom. We also compute E-state indices for the atoms in the molecule. We have some local shape descriptors, notably Sheridan's span descriptors. So these are kind of interesting descriptors. One of them kind of tells you if the compound is kind of on the edge of the molecule or the interior of the molecule. And you can think if it's out on a long alkyl chain, that would be highly accessible to the enzyme. It would kind of go into the active site, whereas if it was on the inside of the molecule, steric interactions might prevent it from getting close to the active site, which requires an oxygen to be transferred from the enzyme to the molecule. Then we have some proprietary descriptors also. So this is just an example of one of the descriptors. This is the charge on each atom in the molecule. Overall, we have 148 local descriptors, and they're excellent for localized properties such as pKa, sites of metabolism, etc. We train our models using artificial neural networks. So in this example here, these blue squares represent the descriptors or the input to the neural network. Each descriptors or inputs multiplied by a weight and then summed up in the neurons, and then each neuron is multiplied by a weight and summed in the output neuron. And so as you're training the model, you adjust these weights iteratively to optimize the performance on the training set. So we have known values, for example, experimental values such as solubility, and we're trying to reduce the error from the predicted solubility to the experimental by adjusting these weights. Furthermore, we go and we not only use one artificial neural network, we take an average of 33 individual neural networks, and that is the overall output of the model. So this is just a slide outlining how we build the models. First, we start with the 2D structures and the experimental data, compute the descriptors. We have a test set that we set aside and sequester. The rest of the molecules would go into a training set, and we would use the neural networks to optimize or train the data, or train the model, and then we would use those results, or we would take those models and apply them in the test set. Artificial neural networks have a tendency to be overtrained, so if we see a model where the results on the training set are much better than the test set, we wouldn't select that particular model. And then this just shows what we're doing with the neural networks, or the ensemble. Okay, so these are just some of the graphs of the observed values versus the predicted values for the pKa, log P, water solubility, and then fraction unbound, blood to plasma concentration ratio, and PEF. So if you look across these top ones, they're very large data sets, and we very accurately fit these parameters. Some of the ones on the bottom are more smaller data sets. We wish we had more data to train these models, and they should look like these. So now getting more specifically into the CYP metabolism, in 2004, Williams et al. Published a paper looking at the major metabolizing enzymes for the top 200 drugs. Over 3/4 of them, or about 3/4 of them, are metabolized by cytochrome P450, a lesser extent of UGTs, and even a smaller percent of esterases. Then for the CYPs, it's broken up into 3A4, that's responsible for metabolizing almost half of the drugs. 2C9 is next. Looks like 2C19 and D6 are about even, and then we have 1A2. So these are the 5 major cytochrome P450 metabolizing isoforms. So with our CYP models, we have kind of a tiered approach. So the first thing we do is we look at a molecule and we say, is it a substrate for 1A2? Yes or no. So we have classification models, and so in this example, 4 out of the 5 classification models said that it was a substrate for that particular CYP. So the only one that wasn't a substrate for was 1A2. And here with this, with that graphic, what this is you're looking at is the length of the wedge here. So this is a short wedge, meaning no, it's not a substrate for 1A2. The rest of them are the longer wedges. For diltiazem, then we will predict the sites of metabolism for each isoform that it's a substrate for, and I only show 3A4. There'd be equivalent predictions for the other 3 isoforms of metabolized that are predicted to be substrates of for this compound. So you can also see these small numbers. These are the probability that kind of the output of the model. The higher the value, the more probable that site is. And what we do is we try and identify the top tier site. So there's kind of a cutoff of what we would determine is a site and what's not a site. And here we've highlighted 4 atoms in the molecule as sites of metabolism. This one, you'll note, is redundant due to their symmetry equivalent, those 2 atoms. Next, we predict kinetic parameters. Here I'm just showing the clearance values, and I have a little bit more on the next slide for KMV max at clearance. But we would predict those for each atomistic side, or each atomic side, and then we would kind of combine those together to get a clearance value for the overall molecule. And I'll show clearance values because it's a simple value, and the higher the value, the faster the clearance. If I showed Km and Vmax, then you kind of have to, it's just harder to pick out 2 numbers or look at 2 numbers rather than 1 single number. And then finally, we would predict the metabolites of the compound. So I'll draw a scheme like this. Now, I mentioned we were using ADMET Predictor 7.2 earlier. This is actually a graphic from the next version of ADMET Predictor. And in this next version, we show the CYPs responsible for the metabolism. So for example, in this upper one, we're oxidizing the sulfur in the ring to create the S oxide, and we're saying that that's due to CYP3A4 and 2D6. Then we also give, if you take each one of the individual clearances and sum them up, it's going to equal the overall clearance of the molecule. This one's 40.1 microliter per minute per milligram of microsomal protein. And then if you sum up the clearances for the individual one divided by the overall clearance, you can get the percent metabolite. So here we're predicting the desmethyl is in 58% of the metabolite. We can also show this as a pie chart. Here's a real pie chart where the width of the slice of the pie, the bigger the slice of the pie, the more the metabolism. So in the liver, 3A4 is the majority, lesser extent from 2D6, 2C19, and 2D6. If you go to the gut, where the expression levels are different, that kind of squeezes out. So 3A4 is expressed in a higher amount than these other isoforms there. And we'll look at those graphics when we look at some of the results of the modeling. We're also predicting, as I mentioned, Km and Vmax. So in this case, you would do an experiment where you would vary the substrate concentration and look at the velocity of the reaction. At a certain concentration of the substrate, you're going to reach an asymptotical limit. That's termed the Vmax. You go down to half of the Vmax and then across to the curve, and that's your Km. So Km is the Michaelis constant. It's a measure of the affinity of the substrate for the enzyme, and it's dependent upon the enzyme substrate complex, not the individual concentrations. Vmax is the maximum metabolic velocity. It's the maximum rate at a fixed enzyme concentration, and it's directly proportional to the enzyme concentration. The intrinsic clearance is the Vmax divided by Km. It's independent of flow. Okay, so when we do the PBPK simulations, this is where we get the blood flow involved here. I already mentioned the units, and for the PBPK modeling, what we actually use are Vmax and Km in those simulations, and this allows us to look at the differences in enzyme expression across the tissues. So when we look at these results, we'll not only be able to look at the oral bioavailability, but we'll be able to look at FDP. We handle the PBPK simulations with a combination of the advanced compartmental absorption and transit model, kind of shown here in this figure. So it models the compound going into the stomach and then working its way down the GI tract. The compound can be in its unreleased, undissolved, or dissolved forms, and then it also, you have differential equations describing the concentration of the compound in these various boxes. Then once it gets into the lumen, we start handling it with physiologically based pharmacokinetic parameters or simulations. And so on this next slide, I show what's defined in the PBPK simulation. So we have various compartments, and each compartment represents a tissue. So we have things like the liver, heart, kidney, brain, etc. Each compartment has a specific volume, blood perfusion rate, enzyme transport and expression levels, and then volumes of lipids and proteins. And this is from literature sources. And then we also calculate the tissue to plasma partition coefficient, so a KP. So a KP gets computed for each one of the different compartments. So now moving on to the results, all the molecules were predicted to be substrates of the CYP associated with their major clearance pathway, so that's a good start. Then 42 of the molecules, the CYP isoform with the highest predicted intrinsic clearance was the same as the major clearance pathway, so we're correctly predicting those in 42 out of the 62 molecules. And in the kind of the summary slide, I'll show where we mispredict those for the 20, what's going on in some of those. Overall, 42 of the 62, or about 70%, were within twofold of the reported oral bioavailability. Now this 42 is not this 42. So these are a few different molecules. There's a lot of overlap, obviously. And then 15% of the molecules had greater than twofold over-predicted oral bioavailability. About 18% had greater than twofold under-predicted oral bioavailability. And twofold is a good rough measurement for PK type predictions. So this is just a scatter plot of the observed values on the y-axis versus the predicted values on the x-axis, and you see here these fairly large error bars for the oral bioavailabilities of the compounds. In the green is highlighted 42, about 70% that are within twofold, and I'll show three examples of these compounds, one kind of a low oral bioavailability, a medium oral bioavailability, and then a high oral bioavailability. Down in the right-hand corner, these are the ones where we over-predicted the oral bioavailability, and then in the upper left-hand corner, these are the ones we under-predicted the oral bioavailability. And I'll show an example from this area and this area. So first, down in the lower right-hand corner, I just kind of looked at these one at a time. I noticed that there's three compounds that were under-predicted that are fairly similar to each other. So these are all three tricyclic antidepressants, and they have this dimethylamino group here that is available in each one of these. So I thought maybe there's some similarity between these. So I got the drug data sheet for dioxapine. The observed value, I think from Goodman-Gilman, is 30%. We predict 73%, and the major clearance pathway is 2D6. So if we look at the drug data sheet, it says it's extensively metabolized by oxidation and demethylation. The primary metabolite is the N-desmethyldoxapine. 2C19 and 2D6 are the major enzymes, and they're to a lesser extent 1A2 and 2C9. So what kind of predictions did we make on these compounds? We predicted that this is hydroxylated in this position, then the seven-membered ring here gets oxidized here, and that forms an unstable intermediate that will fall apart into this compound. The one of the methyl groups gets hydroxylated, that forms an unstable intermediate, which produces the N-desmethyl. So we correctly predicted that the N-desmethyl is the major product here. But if you look at the fraction metabolized, we have 1A2 as the largest percent here. So maybe incorrect on that. So maybe we're just underestimating the 2D6 and 2C19 clearance, and similarly possibly for imapramine and chlorpromazine. Okay, now we move on to an example in the upper left-hand corner. This is galantamine. Here we've underpredicted the oral bioavailability. The observed value is 99%, and we predicted 17%. The experimental major clearance pathway is 2D6. So we create a lot of metabolites of the compound. The major metabolite is shown over here on the left. This is the demethylation of the methoxy group here, and we've predicted a clearance from 1A2 of 70 microliter per minute per milligram of microsomal protein. So this is a pretty high number. If you looked at the previous slide or slides, the clearance for a methoxy group was a lot smaller, so potentially we're overestimating the clearance by 1A2. So now I'll move on and show three examples where we have very accurate predictions. So in this case, the reported number is 99%, and we have a predicted value of 100%. The major clearance pathway is 2C19. And when you look at our predictions, it predicts two metabolites, but very, very low clearance, so 0.8 microliter per minute per milligram of microsomal protein, so a very low number here. Low predicted intrinsic clearance, which is what you'd think of if you had a high oral bioavailability like 99%. The next example is vinorelibin. The observed value is 27%, and we predict 26%, so really accurate. The major clearance pathway is 3A4. We do predict that it's also a substrate for 2D6. Then if you look at some of the a large number of metabolites here, and then I've kind of just in red made this number a little bit bigger so it corresponds to what's highlighted in red. But so you see a lot of clearance due to 3A4. The overall clearance is 153, so high predicted clearance, which you'd expect with a low oral bioavailable compound. The final example is itraconazole. This is an antifungal compound. They're a kind of a large-looking molecule, just to my eye. So here we only predict that it's a 3A4 substrate, and so obviously we got the major clearance pathway correct because we're not seeing it from the other isoforms. So 3 metabolites are generated here. You get some hydroxylation of this isobutyl group over here, one to form this product where it dealkylates this ring, and then this one's just alkylating the carbon on the kind of the outside of the molecule. You get a lot of metabolism from oxidizing or hydroxylating this carbon here, which forms an unstable intermediate and causes this to fall apart into these two. And if you look at these numbers, they're very high clearance values. And this is actually almost an order of magnitude higher than what I showed in the previous slide for vinrelabine. But the oral bioavailability is higher, almost twice as high here, even though the clearance is higher. And the reason being is that the fraction unbound in the tissue is very low for itraconazole, so it's bound more to the tissue and not available for the binding to the cytochrome P450 in order to metabolize it. So overall, in conclusion, our substrate models always predicted that the drug was a substrate of the major clearance pathway, and 42 out of 62, we correctly predicted the highest fraction metabolized was the same as the major clearance pathway, and predictions that are outside of this twofold have lower correctly predicted maximum clearance pathway. So for the 42 compounds we got correct, in 32 or 76% of that, the highest fraction metabolized was the same as the major clearance pathway. If we go down and we look at the ones that were over predicted by greater than twofold, only 66% of those were correctly predicted. And then if we look in the upper left-hand corner, we only get 36% where the fraction metabolized was the same as the major clearance pathway. And then overall, 68% were predicted within twofold error of the reported value. But all of this table basically gets that we're either over-predicting or under-predicting the clearance. Okay, so I'm going to move on to the next point, which is our next section, which is using in silico modeling to predict the unforeseen renal failure caused by SGX-523. So SGX-523 is a quinoline-containing molecule. It was developed by a company called SGX Pharmaceuticals and was one of the first examples of structure-based design, I believe. And it's a kinase inhibitor with an IC50 of 4 nanomolar. It was very selective for other kinases. When they did experiments of xenografts in mice with both lung and gastric cancer cells, it inhibited the growth. So everything was looking good at this stage of the game. They then performed some microsomal studies in rats, dogs, monkeys, and humans, and all the profiles looked similar in the microsomes. So they did some drug-enabling studies in rats and dogs. Then when they got to phase 1 clinical trial, they dosed 6 patients with 80 milligrams of SGX-523. All of them developed renal failure. And so in a report by Diamond et al. In 2010 in Drug Metabolism and Disposition, they determined that the drug-induced neuropathy was due to a metabolite of 523 that was not detected in the preclinical studies. And it turns out that through some good investigation of Diamond et al., that aldehyde oxidase is what's transforming the quinoline ring into a quinolinone ring, and that decreases the solubility. And formation of this is species dependent. So it was formed in human and monkey, but not dog, and that's why, you know, if they would have performed these studies in a monkey, they might have seen this effect and then went back and looked at a different drug. When they went into the clinical trials, I heard a talk by one of the medical officers here that, you know, they had one shot at this clinical trial, and when it failed, the company went under. So a little bit more information on aldehyde oxidase. It's a phase 1 non-P450 cytosolic enzyme, and since it's in the cytosol, you can't, you won't see it in microsomes. So you have to use either the S9 or the cytosolic fraction of the liver cells in order to see this effect. It oxidizes aldehydes to carboxylic acids, but it also oxidizes nitrogen-containing aromatic rings. For example, this quinoxalone ring shown here. And that's the reason it's becoming more important. There's a lot of kinase inhibitors going into clinical trials and passing those, and they contain a lot of nitrogen-containing aromatic rings. The distribution expression is highly species dependent, so it's high in human and monkeys, low in rat, and deficient in dogs, and that's why they didn't detect this in the early stages. An example of a molecule being oxidized or metabolized by aldehyde oxidase is shown here. So it's bromonidine. The aldehyde oxidase would hydroxylate the position adjacent to a nitrogen. This is also a site of metabolism for aldehyde oxidase. It forms this group, which would then tautomerize into the keto form, which is more stable than the enol form. So this is what's going to be observed. So what we did, we're just trying to examine this using in silico modeling. So we generated metabolites of SGX-523. Then we predicted the physicochemical properties of SGX-523 and its metabolites. Then we used those predictions to create a PBPK model to track the M11 metabolite in monkeys, both in IV and PO doses. The IV and PO doses were experimentally performed in the Diamond paper et al. And then we also created PBPK model to track the M11 metabolite in human, and then compared the predicted solubility of M11 to the concentration in the kidney tubule. So this compound is SGX-523. It contains a number of rings, number of aromatic rings here. This is your quinoline ring up in the upper left-hand corner here. The compound is a biopharmaceutical classification system class 2 compound, which means it has low solubility and high permeability. Our predicted log P is 3.04. Experimentally, they measured the basic PK of 3.8. We get a prediction of 4.2. Solubility in water experimentally is 5.4. We predict a solubility of 2.5 micrograms per milliliter, so pretty close to this estimate here, or the experimental value. And then they also looked at solubility in a 1 normal solution of HCl, and the solubility goes way up, goes from 5.4 all the way up to 2.7 milligrams per milliliter. And if you remember from the 62 molecules that I looked at in the previous section, 2 micrograms per milliliter was one of the lowest predicted solubilities. So we're kind of on the low end just with the parent compound. If we look at a plot of the log of the solubility versus pH, you'll see that the solubility starts increasing at low pH when you're forming a lot of ions here. From the studies from the Diamond et al. Paper, they used different fractions, or they looked at microsomes S9 fraction and cytosolic fraction, both with and without NADPH. So I'm just summarizing the results here. The primary metabolites are the N-des-methyl and two monooxygenated species, M6 and M9. Both of these required NADPH and was also present in the microsomes. The 2-quinolinone structure was NADPH independent and only observed in the S9 and cytosol. Then the secondary metabolites were M3 and M4. The one in blue here required both the liver S9 fraction and or appeared in both, had to be produced by both of those. Okay, so now we're only predicting CYP metabolism. So I show the CYP metabolism here. This is SGX-523. We're predicting that the sulfur will be oxidized and then the pyrazole ring demethylated. We also predict that 2C9 hydroxylates in this position and then forms the lactam ring. Of course, this is not quite true. It was actually aldehyde oxidase. And then we can go a second round on each one of these metabolites to produce secondary metabolites. And so this can either be produced from the first step via aldehyde oxidase and then demethylation. So this is our M8 product from the previous slide. M5 was a primary metabolite, and then M3 could be produced either via this route or first demethylating and then oxidizing the sulfur in this position. We're not sure if this is M6 or M9. They just quote a single oxidation species. So even though we predict that this is formed from 2C19, we predict it's produced in a very small amount. So in that extent, I thought we're okay with this particular prediction since it was only produced in a small amount. This next slide shows the physical, chemical, and biopharmaceutical properties of the metabolites. The thing really to emphasize here is the decrease in solubility of the SGX-523 at 2.5. It goes down by quite a bit, down to 0.56 in M11. So this is the metabolite we'll track. I also want to note that M8 is fairly insoluble, at least our predictions indicate that. And we could track this. We didn't do this, but I think this might also be potentially crystallizing in the kidneys. They do note that this does show up in the urine in the experimental procedures. So let's take a look at the microstates for SGX-523 and kind of help explain what it's doing in solution. So there's a pKa at about 4.2 where the quinoline ring gets protonated. As you decrease the pH down, there's another pKa at 2.15, and it's producing a whole slew of microstates that all are formally +2 charged. If you go down even further, you go to the +3 state. So if we look at the fraction ionized versus the pH, here I'm just plotting the macro species, you'll see that between 2 and 4, this one in blue, which is the +3 state, the +2 state, and the +1 state, we're getting a lot of ionization at these lower pH values, and that's responsible for the spike in solubility. If we look at M11, this doesn't have the quinoline ring, it's a lactam ring to protonate, so there's no pKa at 4, but as you get even lower, it does protonate some. So it's fraction ionized versus pH would have a much lower contribution in 2,4 to the +2 and +1 states. So if we graph all these on one graph that's shown here, so this is the predicted solubility versus the pH for SGX-523, the M11 metabolite and M8 metabolite. And so again, what you're seeing here is that when the compound goes in the stomach, it becomes very soluble, and so the dissolution process is very quick. The other two compounds, when you get into the biological pH, have a much lower predicted solubility than SGX-523. Okay, so now we're turning to the PBPK simulations. First, we simulated an IV dose in the monkey, and we used a 1.6 mg per kg. And this is similar. This was the dose used in the experiments from the paper. We don't get a lot of experimental data out there, so we tried to mimic the results from that with the PBPK simulations. Now, when we perform this simulation on the left, we just assume the parent is cleared by aldehyde oxidase. So that was the only enzyme we're looking at here. The paper reported a Vmax of 140 nanomole per minute per milligram of protein for aldehyde oxidase. So we used that parameter, or used that value in our PBPK simulations. We assumed that was an unbound value. We then assumed that the Km is the same as the 2C9 prediction. Now, we had also done a study where we used the Km and we optimized the Vmax value, and we came out with a very similar result as the experiment. We then also used, on the one on the left, the fraction unbound times the glomerular filtration rate to calculate the amount in urine. And this corresponds to this curve here. So the PBPK simulations on the IV dose show that the compound is quickly metabolized over a 24-hour period. The amount in urine is shown in red, increasing here. But this parent compound doesn't end up in the urine very much, and the experimental value is 0.2% of the compound is excreted in the urine, and that corresponds with these very low milligram masses for the amount of the compound in urine. We predicted the volume of distribution to be 6.24 liters. The experimental value is 3.25. Then on the simulation on the right, this is an oral dose of 6.2 mg per kg. In this case, we tried to fit the amount in urine to the experimental point, and we did this by optimizing the clearance, the renal clearance, to try and make it go through that point. Now, we could have spent a lot of time on this, or more time on this, and got this to go right through the middle there. But what this is showing is that when you administer the dose to the monkey, the concentration goes up very rapidly in plasma, indicating that it's very rapidly dissolved and gets into systemic circulation. It is cleared quickly by aldehyde oxidase, and then here where the orange line is the amount of urine, M11 in urine. So we used this optimized parameter here in the next slide, where we went to the human oral dose of 80 milligrams of the compound orally dosed, and the orange, the absorption and dissolution plots are shown in red and blue, and then the orange line is the concentration of M11 in the kidney tubule. So again, we get rapid dissolution going into the systemic circulation. This red line is the amount of dissolved SGX 523, and what happens is it kind of, there's a precipitation event for some of this as it works its way down the GI tract, so it crystallizes and then goes back up as it's slowly dissolved here. The amount in urine shoots up very quickly and follows this orange line. The predicted solubility of M11 is shown across this green line here, so you're getting more the compound's concentration in the kidney tubule than the solubility, which indicates that it'll crash out and form crystals. And so this would be the reason that you're forming these crystals, blocking the kidney tubule, resulting in the renal failure. So overall, SGX 523 is a class 2 BCS compound. It's low solubility, but the presence of ionizable groups at a low pH allows it to dissolve. The ionization of the quinoline ring increases the solubility at low pH. As I just mentioned, the aldehyde oxidase metabolite has much lower predicted solubility than 523, and then the PBPK simulations indicate that an 80 milligram dose leads to a concentration in the kidney of the metabolite that's much higher than its solubility. Thus, the compound will cause renal failure or fallout. So hopefully this presentation gives you a flavor of how our quote QSAR/PBPK marriage can be utilized to assist in risk assessment in humans and animals. And thank you for your time, and I'll be happy to address any questions. Thank you.
Okay, thank you very much, Dr. Lawless. That was a great, wide-ranging presentation. We have one question so far.
All right.
It's from, oh, here's another couple more popping up. So we have from Barun Bhattarai, and he's wondering if EEM Huckle is part of ADMET 7.2 or it was also present in an earlier version.
It was present in earlier versions. Yeah. So it's we had these SIP kinetic properties in earlier versions. I think the original, originally we created these for for PKA and sites of metabolism predictions, and they really increased the accuracy of the models.
Okay. How is the fraction metabolized calculated experimentally, in vitro or in vivo?
Boy, that's a good question. So this is like from the paper where they're looking at the major clearance pathway. We calculate the fraction metabolized in silico. So we just look at the overall clearance of the molecule and then what's associated with the different SIPs. So I can kind of go back to that slide. Experimentally, I think you would use recombinant SIP experiments, or you would use maybe some microsomes and maybe put some blockers of various cytochrome P450 to kind of determine what's the major SIP involved in the metabolizing the compound. Let's see, so the fraction metabolized.
I think it's slide 24.
Okay. Okay, well, yeah, this is an example here. So here, what we're doing is we're taking 3A4 clearance and summing those up and then dividing it by the overall clearance to get this chart over here. So that's what we did in the in silico predictions.
Okay. Okay, we have another question about, is the predictive solubility in vivo in the kidney tubule include protein binding and other factors that can increase in vivo apparent solubility?
Yeah, I think they were just looking at the solubility in urine. So it would have, I don't know what, if there's a lot of lipids in urine. So that's how they experimentally measured the solubility.
Okay.
Is John, do you have any comments on that?
I think that our solubility is predicting an aqueous solubility. So assuming you're taking free-form molecule, placing it into some sort of aqueous buffer environment and measuring what or predicting what the saturated concentration is going to be. And then that was compared to what were some of the concentrations we predicted in the kidney tubules. We're not accounting for any special binding that's taking place there, so it's just literally going to see whether or not the concentrations are exceeding what the solubility was predicted to get some idea about whether precipitation might be taking place.
Okay.
And the next question is about predicted F and observed F%. How large is the difference or mean absolute error?
Yeah, I'm sorry I didn't calculate that. I will go back and calculate that and try and email Paul on what that is.
Okay.
So you'll have Paul Thomas's email address?
Yeah.
Let me just write that down. How large is the mean absolute error? Okay.
Okay, the next one is asking about gut metabolism. I think that's just in general was discussed in the very beginning.
Mm-hmm.
Yeah.
Oh, okay. Gut metabolism. So for these compounds, all the fraction absorbed is very high. I think the lowest fraction absorbed, or at least our predicted value, was about 60%, and I think the average was over 90%. So there's not much metabolism, at least predicted, going on in the gut, or excuse me, in the stomach. So we kind of ruled out compounds that weren't well absorbed in there.
Okay. Okay, here's a question about KM and Vmax. If you don't have any existing information, what are the parameters you can consider? So I guess they're thinking if you only have chemical structure.
Yep, I mean, that's what we did.
Yeah.
So with ADMET Predictor, you just sketch this 2D structure of the molecule in it, push a button, and it'll predict KM, Vmax, and clearance for the 5 major isoforms.
Okay. And I think he has a quick follow-up.
Mm-hmm.
Whether you're considering microsomal enzymes in the gut or other tissues.
Microsomal, which question is this? Oh, this is, in the whole scheme, we are not considering microsomal enzyme trapping in the gut or other tissues. No, let's see, lysosomal, maybe you're talking about lysosomal trapping. The software can look at that, but we weren't looking at that in this particular study. Okay. Lysosomal trapping, yeah.
Okay.
You can't pick that up. So lysosomal trapping is a compound can get into lysosomes, but the pH is different, and it can be ionized, and that prevents it from getting out of the lysosome. And so that can be incorporated in the PBPK simulations, I believe. John can correct me if I'm wrong on that.
No, that's pretty good. I think that by default, we're assuming that there is 100% unbound material in the enterocytes in the gut wall. And we've got some other tools here at Simulations Plus, primarily a tool called Membrane Plus, which does give some information about the impact of lysosomal trapping. And if we can see in that Membrane Plus simulation that lysosomal trapping is going to be important, we could extrapolate some of that information and modify some of the inputs in the enterocytes to help us capture maybe a little bit more accurately what's the unbound fraction and how that might impact metabolism. And then also I think Michael mentioned, with respect to the comment about considering microsomal enzymes, we do have the expression levels of the enzymes set up in the gut and in the various tissues of the PBPK model. So while we do predict 1 Vmax and Km, we are going to be scaling the metabolism rates based upon the expression levels along the gut, and then primarily in the liver tissues if we're talking about CYPs, and some other tissues if we're talking about other enzyme systems.
Yeah, that's very important.
Okay. So we have just a couple more questions. This is a really good one. What's that?
I'm sorry, they're coming in fast and furious. Yeah. Looks like.
So with some in vitro knowledge, so some experimental data, is it possible to change how ADMET Predictor handles metabolism? So in other words, can people customize the model with their own data?
Oh, it, yeah, you would, with the way we calculate these atomic descriptors, and I also might mention some of the Fukui indices, that's kind of proprietary within the company. So if you had KM and Vmax values, we would have to have a collaboration kind of to create those specific models. So we could certainly help you out. It's not, ADMET Predictor does have a module that will create QSAR models, but in the case of KM and Vmax, they're kind of proprietary, or they're descriptors that aren't available to the end user. So we'd have to work together in order to redo those, but we'd be happy to collaborate with you if you had KM and Vmax values. And that was creating this data set of KM and Vmax and clearance values was a lot of work, and I need to acknowledge Jinhua Zhang for doing that. And he spent a lot of time going through the literature, downloading articles, extracting the data, and then, as I mentioned earlier, the curation step of the model building is very important. So especially for 3A4 recombinant SIPs, there are cofactors, and in some of the journal articles, cofactors were omitted, in other articles that were included, and so you had to kind of go, and this would give very different kinetic parameters, so you had to go in there and really look at which one was doing the experiment correctly. Also, with these experiments, you're not looking at the disappearance of the reactant, you're looking at appearance of a particular product. So they're just much more difficult, time-consuming experiments. Sometimes you can do really good experimental work, and when you publish it in the literature, maybe instead of having the OH here, you have the OH here, you incorrectly, just make maybe a typo. So we had to go through and look at a lot of that. A lot of the there were some transcription errors. For example, sometimes I think people would put in micro, a mu that would be converted to an M. So you'd get a really high clearance or a low clearance that was incorrect. So we really had to curate the data quite a bit.
I think this question gets to something that I was also wondering, asking about whether the model is limited by the training set for specific drug types. And so my sort of similar question was, are there areas where you just don't have very good data, so the chemical space isn't very well represented?
Yes, yeah, that can be a problem. Now, on our whole molecule models, we'll flag those, that if the molecule is vastly different than anything in our training set, we'll color that and flag that. In the atomic property predictions, we can't do that because it's kind of a combination of a lot of different models. But when you look at, you're looking at the environment around an atom, and so I think there's a less discrepancy there for particular atomic compounds. So kind of, like a carbon in this phenyl ring, if it's being hydroxylated, it might be a similar, the descriptors might be similar to that if it's in a much larger, maybe an environmental chemical. So I think there's less problem with the atomic descriptors, but certainly there is some out of scope values there.
Okay, so we're going long on our time, and I want to respect our panelists' time. So I'm going to just go ahead and ask one more question, and then we'll write down the rest of the questions and get answers back to you.
Okay.
Are there any advances in prediction of passive permeability to organs in permeability limited models? And this is referring to slide 20.
I'll let John answer that. Okay.
Okay, the short answer is yes. There have been some advances, not so much from a purely in silico perspective, though. So in the GastroPlus PBPK modeling platform, there is a feature called transporter-based IVE, where you could take some in vitro data, if you've measured it in hepatocytes, some coefficients related to uptake into the hepatocytes. Or even more promising is taking some in vivo PK data that you might have from a small animal study and using that to define what we call a specific permeability surface area product, PSTC. And we've been able to show that if we do have a good value, either extrapolated from some in vitro studies or optimized from some in vivo IV PK data, that one parameter allows us to scale all of the individual tissue permeability surface area products based upon differences in cellular volumes. And even more exciting is we've been able to show that if we do use, let's say, some animal IV data to optimize that specific PSTC, that can be then translated across species or go into humans. So we can utilize that animal data, optimize that specific PSTC, and then have a good set of inputs available for estimating those permeability coefficients when we do our human predictions. So there are some advancements, but not yet from just a purely in silico perspective. We still have to utilize some in vitro or potentially limited in vivo PK data.
Okay.
And Christy, I do see one question I can answer quickly. So it's
Okay.
How long will it take? How long will it take to predict oral bioavailability for 100 compounds, and which experimental parameters are needed as inputs?